And good evening and welcome to the 11th season of Basic Science Lights the Way. Thank you all for joining us tonight, whether this is your first Basic Science Lights the Way or your regular attendee. We appreciate your interest in supporting basic science research. It is my hope that this series will spark a renewed sense of connection between fundamental science and the remarkable research and innovations that are shaping the future. It's impossible to predict the potential applications of curiosity-driven research, but that is what makes it so exciting. Fundamental science done at Berkeley has proven to uncover major discoveries that change the way we see the world. Just take CRISPR-Cas9, the laser, the telomerase, and other examples. Even just this month, you may have noticed that the Nobel Prize in Physics was awarded to Emeritus Professor of Physics John Clark for his discovery of macroscopic quantum mechanical tunneling and energy quantization in an electric circuit. The circuit was essentially a qubit, or quantum bit, the basic component of today's prototype quantum computers. This work laid the foundation for exploring macroscopic quantum mechanics in superconducting circuits. Tonight, we'll hear from three faculty members on the frontiers of basic science research: a microscopist peering inside brains, a physicist searching for dark matter, and a biologist studying natural killer cells, all of whom are answering questions we never dreamed of asking in the first place. Before we begin, I want to comment on the current threats we're facing to basic science. Our research faces significant funding cuts nationally and also at Berkeley, at our university. Those of you who want to help can make yourselves heard as a voice for the importance of basic science, which is something our team will continue to do through this series and beyond. For those of you who are philanthropically inclined, I encourage you to reach out to us. We appreciate your prior support, and we are likely to need your help, unfortunately, increasingly over the next few years. With that, I'd like to introduce our moderator for this evening, Kathy Yelick. Kathy is Berkeley's Vice Chancellor for Research and the Robert S. Pepper Distinguished Professor in Electrical Engineering and Computer Sciences. She provides primary leadership in research policy, planning, and administration, including relations between the university and industry. Kathy is also the Robert S. Pepper Distinguished Professor of EECS and a senior faculty scientist at Lawrence Berkeley National Laboratory, where she previously led the NERSC Supercomputing Center. Her research is in high-performance computing, and she's worked on interdisciplinary teams developing scientific applications, including chemistry, fluid flow, genomics, and fusion. Thank you for being here this evening as our moderator, Kathy, and let me turn it over to you.
Well, thank you, Steve. It's great to be here this evening with all of you, and I'm excited to be able to moderate this wonderful set of presentations. As the Chancellor often says, fundamental research is the seat of Berkeley's preeminence. Curiosity-driven, fundamental science advances our basic understanding of how and why things work. Berkeley is fully committed to making these basic research results open to the public, and others can build on that, ask follow-up questions, and use them to train future generations of scientists. Part of Berkeley's core mission is to impart knowledge upon those ready to learn. Why else would we run this series but to continue this lifelong love affair with learning? My own research has been in high-performance computing, designing algorithms that use thousands of processors to understand things like microbes, such as bacteria, viruses, and fungi, and how they're impacted by environmental events, such as wildfires or drought. The original inspiration, by the way, for this work was having somebody tell me that it was impossible to use supercomputers for this problem, that you couldn't run this type of computation on a supercomputer. So with that as kind of inspiration, let us dive into the conversation. And at the end of each talk, we will try to address all the questions from the audience, so please add your questions to the chat. It's my pleasure to introduce our first speaker of the evening, Associate Professor of Physics, Ben Safdi. Ben Safdi's research investigates the microscopic nature of dark matter, which is currently not well understood. He has helped to establish pathways towards potentially discovering some of the most well-motivated dark matter candidates So potential explanations of this dark matter at the microscale, such as axion dark matter, with novel laboratory experiments and astrophysical probes. In addition, he has developed data analysis techniques that have helped better understand how to search for signatures of dark matter in complicated astrophysical data sets. Dr. Safdi also performs calculations and simulations within the context of dark matter models such as axion models, making use of high-performance computing resources that inform these searches. Ben, thank you for joining us this evening. Can you share with us some of your research, please?
Well, thanks a lot. Today I want to tell you why axions could be the next big discovery in physics and what we're doing here at Berkeley to try to find them. Axions are a hypothetical particle of nature which are at the center of some of the biggest questions in science, such as what is the nature of the dark matter in our universe, how does gravity work at the smallest distance scales, and why does the neutron not appear to have an electric dipole moment, which goes under the name of the strong CP problem. Let me tell you a little bit about this problem first. The strong CP problem refers to the absence of a neutron electric dipole moment, but that's pretty complicated, so let's start by looking at water. Water is charge neutral, but it has a charge asymmetry. There's more plus charges on one side, minus charges on the other. And so what that means that if we put water in an electric field, it will rotate in the presence of that electric field because of this charge asymmetry. So now let's zoom in on a nucleus. A nucleus has protons and neutrons. Let's look at a neutron because it looks an awful lot like water. In particular, it's composed of charged constituents, which are quarks, even though it's charge neutral. So we might expect an electric dipole moment, which would cause the neutron to precess in the presence of an electric field. More precisely, the standard model has a fundamental parameter of nature, which we call theta. And this theta parameter, it describes the asymmetry between these quarks within the neutron. So if theta happens to be non-zero, then the neutron would precess in the presence of an electric field. But if theta was exactly zero, then by symmetry, all the quarks would be aligned and the neutron would not precess. Now, a fundamental parameter of nature is something like the electron mass. It's a fixed value of our universe which cannot change. And the strong CP problem is this puzzle that when we look at data, no neutron electric dipole moment has ever been observed. Neutrons do not appear to rotate in the presence of electric fields. In fact, we've constrained this parameter to less than 1 part in 10 billion away from zero. And this is strange because if I were to randomly create a universe, I draw a value of theta, okay, it lands over here, that's not consistent with our universe, I do it again, it lands over here, still not consistent, and I need to repeat this at least 10 billion times to find a version of our universe which looks anything like the universe that we happen to live in. This is an example of what in physics we call a fine-tuning question or fine-tuning problem. And such fine-tuning problems are often an indication that something deeper is going on that we just don't understand. And in the late 1970s, these very smart people proposed such a dynamical explanation. What they noticed is that when we randomly draw a value of this theta parameter, the neutron really wants to push it to zero. That is, the neutron would be in a lower energy configuration if all the quarks could make themselves align. But they can't. It's a fundamental parameter. It cannot change. So what they did was they promoted it to a dynamical field, a dynamical quantity, which is called the axion. Now, since this quantity is dynamical, the quarks can relax to the lowest energy configuration in which they're all aligned, thus explaining why the neutron doesn't have an electric dipole moment. So the picture which emerges from the late 1970s is that perhaps the neutron isn't just composed of quarks and gluons, but they're also this hypothetical axion particle bound up inside of the neutron, whose role it is to cancel all this electric dipole moment. But I should emphasize that this is hypothetical. We don't know if this story is true, and that's what we're trying to figure out. It's also understood now that axions could explain the dark matter in our universe. One of the biggest questions facing modern science at the moment is what is this mysterious dark matter which is out there in our universe? In fact, dark matter is around 5 times more prevalent than the ordinary matter that we're used to. But we don't know at all what this stuff is at a microscopic level, but it could be axions. Understanding how axions can explain the dark matter of our universe is actually a big part of the research in my group. We perform supercomputer simulations using some of the biggest supercomputers in the world here at Berkeley to understand the complicated dynamics, some of which you're seeing here, that could give rise to axions as dark matter being produced in the instance directly after the Big Bang. In addition to explaining dark matter, axions also appear to rise generically from our attempts to construct a consistent theory of gravity at the smallest length scales. Our best theory of gravity at the moment, at the smallest length scales, is a theory called string theory. And axions are a generic prediction of string theory because string theory has two crucial ingredients that give rise to axions. In addition to having the three spatial dimensions that we're used to in time, string theory has extra dimensions, 6 of them, which must be compact, they must be made small. String theory also has forces like gravity and electromagnetism. It turns out whenever you have extra dimensions and you have forces, you also end up with axions. Now the axion has one free parameter, and that is its mass. And at the moment, from data, its mass is constrained only to around 1 part in 10 billion, which is a huge range of possibilities. But earlier this year, my group at Berkeley was able to narrow this mass range down to only 1 in 100 if axions arise from string theory. Now let me tell you why no institution is better suited to discover axions than UC Berkeley, or how we will discover dark matter on the UC Berkeley campus. A few years back, my colleagues and I came up with an idea for a new type of experiment that could discover axions if they're dark matter in precisely the range of masses that we predict axions should exist from string theory. And I want to briefly explain to you how this experiment works. We take a toroidal magnetic field in the laboratory. This is a magnetic field in the laboratory which goes in a circle. What we are able to show is that in the presence of axion dark matter, you end up with an effective electric current which follows that magnetic field line and which oscillates at a specific frequency determined by the mass of the axion. This, by the right-hand rule, induces a real secondary magnetic field which pierces the inside of this toroid and oscillates at this same frequency. We then put in a pickup loop into this cylinder, and as the magnetic field oscillates through magnetic induction, we get a real current, which we can try to measure. Now, we don't know the mass of the axion, but we can tune, just like tuning a radio, we can tune this circuit to scan over many different possible masses, in particular this factor of 100 of masses that we need in order to probe axions from string theory. So we've already built a prototype of this experiment. It was called Abracadabra 10 centimeter. Here you're seeing an image of this detector. It was literally palm size. It could fit within my hand. And I'm highlighting some of the experimentalists from that project, in particular Chiara Salemi, who's actually now a professor in the UC Berkeley physics department. She was the lead graduate student on that experiment at the time. And what Chiara and I are doing at the moment is trying to scale up this prototype detector from 10 centimeters to 1 meter. Because if we can make a 1 meter version of this detector, we predict we'll be able to detect axion dark matter if axions are coming from string theory. We'll be able to detect anything across that entire possible parameter space. So axions are extremely well motivated. They could answer some of the biggest questions that are out there in nature. Tell us about dark matter. Tell us if we live in a world with extra dimensions. And let's make this discovery happen at UC Berkeley. Thank you. Happy to take questions.
Great. Well, thank you very much, Ben. It does look like we have at least one question from the chat. And the question is, what are the predictions for the distribution of the axions within the galaxies, or are they uniformly distributed throughout?
That's a great question. They're actually not uniformly distributed. The axions have a lot of complicated spatial structure. The dark matter would have a lot of complicated spatial structure. I actually showed an image of that earlier in my talk. And in fact, when we think about building blocks of life, we don't normally think about dark matter, but dark matter plays a crucial role because dark matter forms the structure. This is what we understand from the evolution of our universe. Dark matter first forms structure, complicated, compact structure, and then pulls in the ordinary matter, which forms stars. So when you look up in the night sky, you see all of these beautiful stars, galaxies. What you don't see, but we can infer with astrophysical observations, is that hiding behind that is just as complicated a distribution of dark matter, which would be axions, if axions are dark matter. And that structure actually formed first, then pulled in the ordinary matter. Without dark matter, we would not have all that beautiful structure that we see when we look at the night sky.
Great. So you talked about the axions having a free parameter, which is its mass. Can you say more about what will determine this mass?
Yeah, thanks for the question. So the axion I mentioned has one free parameter, which is mass. And you might ask the question, well, where would that come from? And the answer is it's the size of these compact extra dimensions, which I told you about. So if those compact dimensions are bigger, then it turns out that the mass goes up. If they're smaller, that mass goes down. And what string theory tells us is it gives us a preferred size for those compact extra dimensions, which points us towards this relatively narrow range of masses.
Great. You also talked about how you think Berkeley is going to be the place where this discovery or these discoveries are going to be made. Can you say a little bit more about the competition? Where is the other work going on to study axions?
So there's a worldwide search to discover the axion. This is universally or almost universally accepted as our best candidate for dark matter at the moment. But there's a huge possible range of masses. And much of the experimental effort is actually happening outside of this mass range, which was targeted for string theory. And that's because it was only relatively recently that my colleagues and myself and others were able to come up with these new ideas to search for axions in this most motivated range. So it's only been a few years now that we've actually had the ideas for how to look for axions in their most motivated region of parameter space. And at the moment, Berkeley has a huge advantage because the one experiment which has operated in this range, abracadabra, many of the lead people on that experiment are here in Berkeley, myself, Chiara, but it's not just us. Everyone is trying to gear up to solve, to find axions. Our colleagues at Stanford are doing the exact same thing, just in the southern part of the Bay. So, and our colleagues in Europe also. And of course, we're all collaborating together, but friendly competition is also useful.
Great. Well, let's see. So we have a couple of other questions from the audience. So one of them is, you described the axion as a solution to the strong CP problem by being something like a portion of the quark alignment probability landscape. Is that a correct sort of assessment or summary? And can you make that kind of axion identity any clearer?
Yeah, so the way that you should think about the axion is you should think of it as this angle between the quarks within the neutron. And without the axion, the version of the standard model that we have at the moment, the standard model being the accepted model of particle physics, which is the most precisely tested theory ever in humanity. At the moment, that theory has a fixed parameter which rigidly determines the angle between the quarks and the neutron. And that theory just appears to run into problems with data, which is that all of the quarks are extremely well aligned. What the axion does is it makes that parameter dynamical. It removes its rigidity so that it can relax down to its lowest energy configuration where all of the quarks are aligned. So you can think of it indeed as an excitation associated with changing the angle between the quarks within the neutron.
So I think that that partly answers the next question, which is about whether an axion is really a fundamental particle, because you've been talking about this sort of structure of the axion itself as if it's not just sort of, I don't know, something that, kind of some fundamental particle. So can you say anything about that?
Yeah, so an axion is a really fascinating type of particle because in particle physics language, it's a boson. And there's actually only one other boson we know about in nature, which is the Higgs boson, which many of you might have heard of. This was a big discovery to come out of the Large Hadron Collider at CERN in Geneva. And the Higgs boson is also a strange type of particle because we use different words to talk about it. So maybe if you followed this discovery in the news, you would have heard that the Higgs boson gives everything mass. It is the particle which is responsible for giving all other particles mass. At the same time, it's a real particle. We produce it every day at the Large Hadron Collider, thousands of them. And so how can a particle be both a fundamental object and be responsible for some collective behavior? In that case, giving all of the particles mass, or in the case of the axion, making this angle dynamical. And bosons are uniquely suited to do these sorts of collective behaviors. They're able to come together collectively, a large ensemble of them, and affect physics at a really macroscopic level. And that at a really deep level is what's going on with this axion. It is a particle, but it is also responsible for this collective behavior within the neutron.
Great. Well, thanks very much, Ben. I'm all for collective working together to solve big problems. So sounds like that's what these axions are doing too. So or at least to have a big effect. So thanks very much.
Thank you.
And we'll have a few more minutes at the end for more questions if we have them. But now I'd like to move on to our next speaker of the evening, Professor of Neuroscience and Luis Alvarez Memorial Chair in Experimental Physics, Naji. Using concepts from physics, once again, Naji develops the next generation of optical microscopy methods for understanding the brain at higher resolution, at greater depth, and at faster timescales. Besides inventing methods that make an immediate impact on neuroscience, Naji also aims to extend the applications of our technologies to other living and non-living systems. Na, thank you for joining us, and please tell us about your work.
Thank you very much for the opportunity. Can you see my screen?
I can, it's not yet, there we go.
Okay, great. So today I'm going to talk about the brain. So brain is where all our mental activity occurs. Human brain has 100 billion neurons, making about 100 trillion connections, where they communicate with each other through electrical activity lasts only about 1 millisecond. Even though modern neuroscience started decades ago, the complexity of the brain leaves many unanswered questions in both basic neuroscience, how the brain works, and as well as translational neuroscience when the brain starts to have diseases. As a joint faculty between physics and neuroscience, my lab used physics concept to invent new microscopy to better image the brain activity, and especially in this case, the electrical activity in the living brain. So the most common way of imaging the brain is to use the small mirrors called galvo mirrors to scan a laser focus line by line across the brain and record the fluorescence coming back at each position. Those galvo mirrors scans tens of thousands of lines per second, which means that we can image, in this example, blood vessels with a fluorescent plasma at 15 frames per second. So you can see at this frame rate, the size of the vessel actually changes with time, but we cannot observe blood flow directly. It is also too slow to image electrical activity directly, which because of the millisecond duration of the electrical signal require us to scan the laser focus at around 1,000 times in 2D per second. So our solution is really to be inspired by art. So this is a picture taken inside a so-called infinity mirror room. This is a very mesmerizing picture, but it was generated by placing a single lamp between a few mirrors arranged at an angle. So this kind of mirrors are called infinity mirrors. And when we place two mirrors at a slight angle, we will have an infinity mirror pair, which generate infinite number of images for each of the small LED lights that you can see in this picture. So we built an infinity mirror pair, which we call facet, for a brain imaging microscope. And what does this pair do is that when a laser pulse enters this facet, it will get converted into a series of pulses that are delayed in time. So they arrive in the brain at different time. They also arrive at different locations. So in other words, the facet mirror pair convert a single laser pulse into a line scan. So a laser that can output 1 million pulses per second will generate 1 million line scans per second, which makes it possible for us to image at 1,000 frames per second. So using this microscope, now we can watch individual blood cells, which are not fluorescent. So they show up in this video as these dark objects zipping through the vessel. But if we image the same cells at 100 frames per second as shown in the video below, it is simply too slow to capture the movement of those cells. So in other words, the ability to track individual blood cells now allow us to map how the blood flow actually vary over space and time. So inside this horizontal vessel, you can see that the flow speed is highest along the central axis. And the cells will slow down when they reach the junction where these multiple vessels converge, just like cars in a traffic jam. And in this small artery, you can also see how the blood flow speed actually change with time rhythmically. So about every 100 milliseconds, the blood flow speed cycles from high speed, as indicated by the red color, to low and back to high speed again. This was caused by the heartbeat of the animal, and every time when the heart beats, it will squeeze out the blood and increase the blood flow speed. Now, with this kind of method, we can now start to image the so-called calcium activity in the brain, which conventional microscope can image a single or at most a few in 2D plans. With the facet, we can image 40 plans simultaneously, as in this example. So hopefully, yes, now you can see all 40 plans, and the small dots that become brighter are individual neurons that become active when we show the mouse an image that those neurons prefer. Here are the zoomed-in view of a few selected plans. With this very high spatial resolution of our microscope, we can measure the property of about 12,000 neurons in this volume simultaneously. Now, this calcium imaging example is the most common approach for activity imaging because calcium signal is slow, therefore it is possible to image with conventional microscope, but it is an indirect measure of neural activity. So to truly really measure the electrical activity directly, or as I call it, imaging the brain at the speed of thought, we need to be able to image at millisecond resolution, which we can now finally do. So here is an image of hundreds of neurons. So each of the small donuts you see are the cell body of individual neuron, but now they are expressing a voltage protein, a fluorescence protein whose brightness is directly related to the membrane potential. So now if you look at the black ticks, there's a super small black ticks. On top of those example voltage traces, this ticks indicates the so-called action potential, which are the electrical signals that last for about 1 millisecond. And the orange trace that is underlying the raw data are the so-called subthreshold activity. This is a type of activity that only electrical activity recording can capture. Calcium imaging will not be able to give you this. So now we can image these activities from hundreds of neurons simultaneously, and this would allow us to answer many fundamental questions on how the brain processes information. But before I end, I also want to show you one example of how this technique can help translational neuroscience by imaging electrical activity in the diseased brain. For example, during epilepsy, which is a chronic disease where the neuron becomes too active. About 3 million people in the US have epilepsy, and 1 million of them have a form of epilepsy that is drug resistant, meaning there's no good medicine to treat it, because their disease mechanism is still not very well understood. So we were able to, using this method, image the electrical activity of mouse brains undergoing epileptic seizure. So the voltage imaging shows that, hopefully you can see that all the neurons, they will get brighter altogether, which means that all of them are becoming active. And then you will see this waves, hopefully you can see from one corner of the image propagating towards the other, showing this abnormally high membrane potential that is propagating during this seizure. So this event has never been observed before, and now we are working with collaborators, try to understand how different cell types, different type of cells in the brain behave and get affected by the epilepsy. So in summary, I just want to, I told you a quick story of how we can use physics to help us to understand neuroscience. So in general, imaging brain at better resolution and speed really requires the collaboration of physicists, biologists, and engineers. And Berkeley is really the perfect environment for pushing the ability of basic sciences to lead the way of our understanding of how the brain works in health and in disease. So thank you very much for your support to Berkeley, and I'm happy to answer any question that you may have.
Well, thank you very much, Na. This is amazing to see the kind of level of resolution that you're able to get from these techniques, and to hear about the maybe improved understanding of epilepsy. Can you use this for other diseases like strokes, this kind of technique?
Absolutely. So with strokes, the particular example I was showing of imaging blood flow would be very powerful. So traditionally, again, the way people try to understand stroke is to introduce a small stroke in the mouse brain, and then you try to look at how the blood flow will be affected. So now we have a way to really look at it at much faster resolution. We will be able to see how individual cells are affected. And in addition, we can combine this blood flow measurement together with electrical activity. So we can not only looking at how blood flow is disrupted by stroke, we can also see how that actually impacts the neurons that are nearby, whether this is to understand how this death of neuronal death occurs as a consequence of stroke.
Fascinating. So, well, thanks very much. And I'll just encourage people to put questions if they want into the chat, but I certainly have some other questions as well. So I think, let's see, one of the questions that has come in is, do you have any insights to the role of the glial cells in epileptic events from this?
Great question. And indeed, glia, there's two main cell type in the brain. One is neurons, which neurobiologists tend to obsess over. But glia is really the support cells. And many, especially neurodegenerative disease, as well as epilepsy, glias are very much involved. So the collaborator that I put on my slide earlier, Jan Paz's lab, are really experts in understanding glia. So we are collaborating together. Actually, thanks to an internal fund on epilepsy, we're collaborating of using voltage imaging to understand both glia activity and neuroactivity simultaneously. Great question.
Great. And what about other organisms? Like I know fruit flies are used a lot as model organisms for things. So can you use this for things like fruit flies?
Yes, absolutely. So the microscopy technique is a very general tool that we can use it to study many different organisms. So we are building up a collaboration with neurobiologists who use fruit fly as a model. Their brain is much smaller. So we could actually do this kind of imaging over a big proportion of the brain, if not the whole brain. And we are looking forward to make discovery using those modern model organisms as well. Kathy, you're
Sorry, I
muted.
was just muted. Can you say a little bit about whether your research relates to mental health and the effects that it has on the brain, or is kind of the structural issues in the brain and dynamics of the brain and mental health?
Absolutely. So many mental health problems oftentimes arise from an imbalance of the chemicals inside the brain. For example, dopamine. My lab doesn't directly work with those problems, but the technique we develop can be used to answer those questions. But at this moment, it's more using animal models because we require fluorescent signal, and there are many good fluorescent sensors that were developed in the past 5 to 10 years that actually allows us to use a microscope to look at neurochemicals like dopamine. So those are the fluorescence protein that they will change their brightness when there's a dopamine release. And the part of the reason that it's very difficult to treat to treat mental diseases is again because we don't really understand all the different chemicals, their roles in the brain, and when brain goes haywire, whether it's through addiction, depression, or Parkinson's disease, exactly how they impact different cell type. So with the imaging method and the new sensors, we will have a very kind of what we call mechanistic way to understand the diseases.
Great. Well, I think we have time for one more question. This one is the sort of two parts. So the question is about whether you've studied other diseases such as autism, and do you think that using the kind of data that you're collecting, could one build a machine learning model of various diseases that you could use for, I guess, better diagnosis and things?
Great question. So my lab hasn't imaged autism models, but we have colleagues in neuroscience department, Dan Feldman's group, study autism. We actually have a joint graduate student. So once we get the current project out of the way, it will be very interesting to look at autistic brain model. The second question is, sorry, can you remind me, Kathy, the second portion of
Well, it's really about building a machine learning model
Yes,
based on the kind of
yes.
data you're collecting.
Another great question. So we are also collaborating with another lab in Geneva Research Campus. So what they do is they image many neurons with a calcium imaging method, and then try to use that to try to understand the machine learning model. So what we're trying to do is to take similar data, but with voltage imaging, and we're able to provide a lot more information than the traditional technique. So this method is very general. So we really expect it to impact both fundamental basic science to applied sciences like machine learning.
Great. Well, this is fascinating work. Thank you very much. In the interest of time, I think we should move on to our final speaker of this evening, Professor and the Esther and Wendy Sheckman Chair in Basic Cancer Biology, David Raulet. David Raulet's laboratory is dedicated to understanding recognition by natural killer cells and T cells, and is developing approaches to mobilize these cells therapeutically against cancer. His lab investigates the mechanisms by which tumors and infectious agents naturally activate or inhibit productive responses by cells. Their aim is to harness these findings to improve existing immunotherapies for cancer and to design new ones. Because of this work, David is a world-renowned expert in cancer immunology and natural killer cells and T cell biology. David, please tell us about your work.
All right, so I wanted to tell you today about T cells and natural killer cells in the fight against cancer and cancer immunology at UC Berkeley. And by way of introduction, I think you know that chemotherapy drugs and targeted drugs act on cancer cells directly. They can cause tumor shrinkage, but they're often not effective in giving cures. I'll contrast that with immunotherapy drugs, which treat the immune system, not the cancer, and they turn out to be more effective on more types of cancer. I'll show you the cells here, the culprits. Here we have killer T cells or natural killer cells. They recognize these tumor cells that are in a dye so that they turn red when they die and they shrivel up. And then these killer cells can go on to kill additional tumor cells or virus-infected cells, for example. So I want to take you back to the early 1990s when my lab and Jim Allison's lab began to ask questions. How do you turn these killer T cells on and, for that matter, off? And so it started with the issue of how you pre-activate killer T cells in order to become effective at killing tumor cells. And we knew at the time that they express these cell surface receptor proteins, and they recognize altered proteins on the surface of tumor cells and other cells. And that provides a key signal, but it was clear it wasn't a sufficient signal to pre-activate the T cells. We knew there was a second signal, but we didn't know what it was. And the work we did together showed that that protein was the CD28 receptor on T cells, what's called a co-stimulatory or co-activating receptor. So this was a fundamental finding about how T cells work. And Jim took it further by investigating a close relative of the CD28 protein called CTLA4. And he expected that it would probably do something similar to CD28, but as it turned out, it had quite a different activity. It actually inhibited T cell activation. So this was quite a remarkable finding, and it led him to ask a very important question, which is whether inhibition by CTLA4 prevents T cells from killing cancer cells. And if that's the case, he reasoned, perhaps we can block CTLA4 with an antibody to unleash T cells to kill cancer cells. So this is what that would look like, blocking the receptor, preventing the inhibitory signal. In 1996, they showed that these injections of antibodies that block CTLA4 cure cancer in mice. This led to a frenzy of subsequent work and finally clinical trials in human cancer. And this is an example in metastatic melanoma, in which at that time chemotherapy drugs were generally ineffective. But with injections of CTLA4 antibody, 20 to 40% of the patients survived more than 10 years and essentially appear to be cured. A related antibody against a highly related inhibitory receptor on T cells called PD-1 were even more effective, and the combination was more effective yet. It turns out this approach, which is called checkpoint therapy, works in varying degrees for more than 20 kinds of cancer, and it turns out really to be the most effective cancer treatment approach in 50 years. So it illustrates how this basic research leads to these important findings. And for this work, Jim Allison and Tasuku Hanjo were awarded the 2018 Nobel Prize in Medicine. But there's some issues. Although it works great, it's amazing, there are some vulnerabilities, and one is that cancer recurrence can occur in many patients with checkpoint therapy. An example is shown here. This is the percent of patients in which tumors initially shrink for several different kinds of cancer with this kind of therapy. And this is the fraction of patients in which cancers recur. And it turns out when they recur, they're resistant to the same therapy. And why do they become resistant, and what can we do about it? Well, we know that T cells, when they recognize tumor cells, they need to recognize these altered proteins in the cells when they're presented by proteins that are on all of our cells called MHC proteins. These are presenting molecules. And it turns out that mutations can occur in tumor cells where MHC is lost, and then the tumor cells become essentially invisible to T cells. And as a result, they can grow out and represent these recurrent tumors that frequently occur. So what can we do about that? Well, I want to introduce you to this other kind of immune cell called the natural killer cell, which has been a major subject in my lab in recent years. And they have a special ability to kill these tumor escape variants. When we started working on them in 1990, they were very mysterious. We knew they killed cancer cells, but we had no idea how. In 1991, we discovered that NK cells selectively kill cells that lose MHC. These are the ones that escape from T cells, in fact. But how do they do it? It took 10, 20, 30 years of research to fully discover all the NK receptors and regulatory molecules present, and here are many of them shown here. They started out naked, now they're very complicated. But some of the receptors, it turned out, were very critical in explaining this ability to kill MHC-deficient cells. Some of these receptors were inhibitory receptors that actually recognize these MHC proteins, and they prevent NK cells from killing the tumor cells. When MHC is lost by mutation, the tumor cells then become sensitive to killing because you've removed inhibition, and NK cells can kill them. So at this point, we knew that NK cells can recognize and kill these escaped tumor cells, but they often fail. And the question is why? Well, we learned more recently that they often fail because they don't receive all the signals they need to sustain their functional activity. Put simply, immune cells often start out as these quiescent cells, and they require activation signals to become fully active killer cells and to sustain their killing activity. And those signals are often missing from tumors. The signals involved include cytokines, which are hormone-like proteins that stimulate immune cells, but also other small molecules that activate immune cells. So can we provide these innate signals, as they're called, to activate the responses? So we benefited from work from our colleagues here at Berkeley, Russell Vance and Dan Portnoy, who had shown that certain small molecules called cyclic dinucleotides activate exactly the kind of response that we would like. And indeed, we went on to show they help activate NK cells to kill tumor cells. We also use cytokine variants, which are called superkines. They're engineered to sustain killer cell activity. And one great virtue of both of these signals is that they also activate T cells. And it turns out that we showed they work much better together than separately. So this shows a proof of principle where we initiate cancer in mice in a colorectal model, where we use a mixture of tumors that express MHC and ones that don't. The tumors will grow out. The checkpoint therapy in this case doesn't work, and the reason we can show is because the MHC-deficient tumor cells grow out. However, the mixture of innate signals that I mentioned do provide strong therapeutic effects in this scenario and in many other scenarios we've studied. And we can show that the NK cells kill the MHC-deficient tumor cells, and the T cells kill the MHC-positive tumor cells. So we think the most effective therapies stimulate both T cells and NK cells. Well, with that in mind, we're working now on an alternative approach to prevent acquired resistance. It involves a cell type called T regulatory cells, or Tregs. They suppress T cell responses, and by coincidence, the discovery of these cells was just rewarded with the Nobel Prize in Medicine a few weeks ago. So our colleague Michael Dupage is an expert in Tregs, and we collaborated recently to show that Tregs also suppress NK cell killing of cancer cells. And we further showed that when you deplete Tregs selectively within tumors, you unleash both NK cell and T cell responses against tumors without causing autoimmunity. And this shows an example here where when we deplete Tregs, we get nice control of tumors that lack MHC. And when we do that with MHC-positive tumors, we also see strong control of the tumors, as shown by work in Michael's lab. So again, we're inducing these mixed NK and T cell responses that we think are necessary to prevent acquired resistance. So to summarize, the cancer immunotherapy revolution actually started at Berkeley. We may not have a medical school, but we nevertheless do important biomedical research here. More than 1 million patients have experienced significant clinical benefit from checkpoint therapy. And we're now working with our new basic understanding of how these immune cells work to make immunotherapy better and improve outcomes for the remaining patients at which immunotherapy is not currently effective. So thank you, and I'd be happy to take any questions.
Great. Well, thank you very much, David. And it's amazing, and I think all of us probably know somebody with that's been treated with cancer immunotherapy drugs. If certainly we know somebody with cancer recently. So the work is really interesting to understand some of the fundamental science behind this. I guess you talked a little bit about how these NK cells are help becoming that sometimes they become inactive within the tumors. And I'm wondering if there's other reasons beyond the absence of these innate signals or the actions of these Tregs that you talked about that cause them to become inactive.
Yeah, indeed, there is. That's a great question. So it turns out that we've recently been been looking at molecules that are expressed within the NK cells themselves that essentially repress the response after the cells have been persistently stimulated for a long time. And we think they play a role in preventing an effective response as well. And we're looking at ways to either drug those molecules to prevent their action, or in a cellular therapy to knock the genes out for those proteins so that we can get more effective immunotherapy.
Great. And can you say something about how are these NK cells kind of looking for cancer cells? What differentiates them that allows it to target it from relative to a normal cell?
A very good question. Yeah, we've done research for years trying to ask what inhibits them, but also what activates them. And that's a critical question here. And what we've discovered is that it's really stress responses that occur in cells, and tumor cells undergo a lot of stress because of their rapid replication and uncontrolled replication that causes the display on the surface of tumor cells of molecules that NK cells can recognize and which activate them. So that's really another point of control of NK cell reactivity, and it's another target that we've been working with to try and improve immunotherapy.
Great. And let's see, can you say something about whether this works for all kinds of cancers, or is it very specific, specific kind of cancer, or does it have to be tailored to the cancer?
Well, it works. This is, of course, still an experimental approach, but it works in our animal models. It works on every type of cancer that we've tested. And we've tested not only, what we call tumor transplant models, where we transfer tumor cells into a mouse that doesn't have cancer, but it also works in primary cancers that we've generated using, for example, carcinogens in these mouse models. The reason that we do that is because we want to mimic real cancer as much as possible, and generally real cancer that starts from carcinogens or other mechanisms are harder to cure than some of the models that people use. But we've seen very nice effects in those models as well. So we're not, as far as we know, this approach may not be restricted by cancer type, but of course we've not tested every type of cancer possible. We've tested probably 4 or 5 different kinds of cancer models, however, cancer types.
Well, great. So let's see. One of the questions is about these superkines, and can you say a little bit more about their structure, a little bit more detail about superkines?
Yes. Well, I have to acknowledge that the superkines we used were actually made by a collaborator with whom we cooperated. He's at the dreaded Stanford University, but these molecules are selected to have different affinities for the different subunits of the cytokine receptors. And it turns out when you do that, you can generate versions that are more effective than the native versions, and can sustain the responses. We also include changes to the protein that extend the half-life of the cytokine once it's injected into the person or animal. Those are critical as well, because cytokines actually normally have very short half-lives in animals, and so we need to sustain them in order to have these positive effects.
And now you've been talking about fundamental science and these scientific discovery in these mouse models. Are there any human trials being planned or anticipated here?
Yeah, there have been some trials. One of the difficulties is, though, that we find that the most effective approach is combining these agents that I talked about.
Mhm.
And so, for example, in the first part, combining those innate stimulators. And it's always much harder to do clinical trials on a combination than it is on the separate agents. And so, and it's a big barrier to get to clinical trials. But yeah, I think we definitely intend to try and get that done. But we don't do that at Berkeley. We obviously don't have a medical school, and in general, the clinical trials will ultimately require investment, usually from a pharmaceutical company, in order to afford them. They're very, very, very expensive types of studies. But yeah, I mean, many trials are being done now on different versions of these molecules, and we think, based on our work, we think that combinations will ultimately be tested.
Oh, what a very, very interesting work. So thanks very much. I think we're going to go back to the full group now, and there's still some other questions that have come up. Some of them specific to each individual, but there's a couple of questions that have come in about the federal funding situation and whether that has impacted your work at all or is having impacts on you or your research group. So I'll just say, as Vice Chancellor for Research, that we see, one of the things that's been in the news a lot is terminated awards. So there's a lot of publicity around, and we certainly have had a large number of terminated awards, some of which have been reinstated because of legal actions. But the other thing that we're seeing is just that the federal agencies didn't spend all of their money last year, even though it was appropriated by Congress. And so I think right now our Department of Energy funding, compared between FY25 and FY24, the previous year, is down by about 33%, which is the kind Department of Energy funds is the number one funder actually of physical sciences work of the kind that that Ben talked about. The other agency that's down is HHS, which of course is predominantly, at least at this campus, NIH, and that's down by about 18%. And so I guess I'm wondering if each of you can say anything about whether you feel like your work is has it been impacted or under threat, recognizing that sometimes you can't quite tell why you might not have gotten a funding renewal or something, but for these things. So we'll start with Ben and then go around the circle.
Yeah, it's a great question, and the answer is absolutely. So we came up with this new idea to discover dark matter, and this idea immediately raised attention in the field, and the DOE, the U.S. Department of Energy, got behind this and essentially committed to building a sequence of detectors under the name dark matter radio. The first of which was supposed to cost $20 million. And that is now postponed indefinitely. It's a huge setback for us because, I believe this is the sequence of experiments which will discover dark matter. So do many of my colleagues. And it's really set us scrambling. We talk to our colleagues in Europe and they say, wait, but we thought you guys were doing that. And we're like, well, can you do it? And everyone is kind of scrambling. It's a huge setback. And we're trying to figure out how to move past it, how to still accomplish this science in this funding environment, but it's difficult.
Okay, and let's see, David, do you want to say anything about this? And then we'll go to Na.
Yeah, I mean, we've seen in, the National Cancer Institute, for example, was funding grants last year to about 8%. About 8% of grants were being funded on initial applications, and that was bad enough. I mean, we considered that really onerous, and it's really dropped to 4% most recently. So we were hearing that colleagues who had grants with 6% of them are not getting funded, at least initially. So that I think is a disaster for cancer. But I think that's just across the board. NIH is really struggling to support people. They're not doing it, and I think there's been a lot of other negative consequences too. The students, what I've seen recently is that because everyone is so concerned about funding, students are having a harder time getting placements as postdoctoral fellows because the labs are, the people who are in labs are not leaving them because they're worried about being able to move to other jobs, or so it's really having a terrible consequence. And that doesn't even speak to the problems with the indirect cost rate, which I'm sure, Kathy, you have to deal with and think about all the time. And it's upcoming.
I won't talk about that one, but thank you. Na, how about you?
So I second what David said. The bioscience research funding is really severely impacted. And as David alluded to, is this uncertainty with the government shutdown, the delay of reviewing the grants that we submitted. This uncertainty is just going to damage science, for years to come, even if we resolve this issue right now. but all the pieces are not falling in places. It's a pretty bad time to be doing science in the US, I have to say.
Yeah, and there's a related question about whether people are leaving Berkeley. I mean, I don't think we've seen kind of a mass exodus at the faculty level, but we certainly are, we have heard of people that don't want to come because they would rather work in other parts of the world. And, it makes it harder to recruit people. And just for all kinds of issues around maybe coming to the US. Especially if you're not a US citizen, but also with the funding issues here in the US, as well as I think what you talked about, David, is probably one of the biggest long-term concerns is sort of the pipeline just getting really blocked because I think graduate admissions are down significantly. We did not at Berkeley unadmit any students last year. I think so we felt we had committed to those students who were being admitted, but as a result this year, I think as we look forward to admissions, a lot of departments there's not a kind of overall university decision to reduce admissions, but it happens naturally because if departments don't feel like they can support the students, then they're not going to admit as many students. And so, and as you said, the postdoc issue also sort of at all different levels, things are being backed up a bit. So, well, I mean, I hate to end with that kind of negative note, but I have to say there's still amazing science going on here at Berkeley, and I think that amazing science is going to continue. And we really hope that we will, first of all, be able to. I think there's a lot of support in Congress for continuing this kind of work, and so I hope to see that going forward, and hopefully we'll see some other opportunities for funding from foundations and philanthropy as well as industry. I know there's a lot of encouragement to work with industry as well. So I think with that, let me just see if I will hand it back now to Dean Harland, I think, who's going to make some closing remarks, maybe somewhat quickly. Sorry.
Well, thank you, Kathy, for moderating, and thanks to all the speakers for their beautiful and compelling presentations. It was really great to see not only what wonderful research is going on, but particularly that it's going on here at Berkeley. So for all of you, we'd like to invite you to join us for the next session on Thursday, November 6th, where members of the Innovative Genomics Institute will be discussing the last decade of CRISPR technology and how they're developing the next generation of genome engineering tools to treat currently incurable human diseases, but also to create sustainable solutions for agriculture and food security in the face of a rapidly changing climate. We hope you've enjoyed this session of Basic Science Lights the Way and learned something new. Thank you for being part of our efforts to lift up scientific research and education, connect curious people to science and ideas, support discoveries of big and small, and particularly thank you for your excellent questions. So please reach out if you'd like to learn anything more about anything we covered. As always, a video of today's session will be available on our website approximately 2 weeks from now. And you can always visit the website to watch episodes you missed. So finally, we hope to see you next season. And until then, stay curious, fiat lux, and of course, go Bears.